Hi everybody, welcome back again. So today we're going to be talking about cell division, but before we can talk about cell division, we need to go over two very distinct groups of cells in the body. All right, so here we have a tree diagram here, all of the cells in the body, and the first branch is giving way to the germline. The germline is found within the reproductive organs and is dedicated to producing a very special type of cell called a gamete. You are probably more familiar with human gametes being referred to as the sperm and the egg. From now on, you also have to be comfortable with referring to these cells as gametes. And then the next branch gives way to the somatic cells. Somatic cells are the other approximately 100 trillion cells that make up your body. The soma in somatic cells means body. The overwhelming majority of cells in your body are somatic cells. We will begin our review of cell division by referring to somatic cells. It will be specified when we begin discussing germ cells of the germline. Germ cells are a relatively small and isolated population of cells located within the female and male gonads. All right, and so here we have the male gonads, the testes within the scrotum, and then here we have the female gonads, the ovaries. In the ovaries, the germ cells are called primary oocytes. So somewhere within these structures, we have germ cells referred to as primary oocytes. All right, and in the testes, the germ cells are called the primary spermatocytes. All right, so the germ cells here's in the male gonads, the testes, are the primary spermatocytes. In the female, the primary oocytes will eventually give rise to a gamete, commonly referred to as the egg or the ovum. In the male, the primary spermatocytes will eventually give rise to the gamete, commonly referred to as sperm. In contrast, the overwhelming majority of cells in your body are the somatic cells. Okay, these are all the other cells except for the germ cells that we just talked about. All right, so we go back to this original tree diagram here. And we have the two major types of cells that we went over, the germ cells and the somatic cells. Now we are going to convert this same schematic so that is in terms of cell division. Germ cells and somatic cells exhibit two different types of cell division. The germ cells undergo a series of cell divisions known as meiosis. All right. And the somatic cells undergo a type of cell division known as mitosis. Therefore, meiosis occurs in the germ cells, and mitosis occurs in the somatic cells. If this is a new concept, write that sentence down and say it out loud at least 4 times over again, or write it out too if it helps. All right, so meiosis occurs in germ cells, mitosis occurs in somatic cells. Before we explore mitosis and meiosis, there is one important set of terms that we have to go over. Diploid, otherwise designated as 2N, versus haploid, designated otherwise as 1N. First, let's review what is meant by the term N. N is the number of types of chromosomes. All right. Recall that humans have 22 different sets or pairs. All right, remember in a pair, there's always two of something. All right, in this case, we're talking about two sets, or rather a pair of two chromosomes, or a set of two chromosomes. All right. So there's 22 different pairs of chromosomes referred to as autosomes, and one set or pair of sex chromosomes. All right, so the autosomes are the type of chromosome, and the sex chromosomes are also another designation for a specific type of chromosome. All right, within all of these specific types, all right, for example, within the specific different 22 autosomes, there are pairs of chromosomes, and within the specific one set or pair of sex chromosomes, there are two chromosomes. All right, but the N is only referring to the number of types. Okay, in this case, we have 22 different sets of autosomes and one set of sex chromosomes. All right, being the basic organizing principle of the genome that is present within the nuclei. So if we have 22 different distinct types of autosomes with a pair for each distinct type of autosomes, and then one set, okay, of sex chromosomes, all right, with a pair or two chromosomes being categorized as sex chromosomes, we take one type of chromosome plus 22 other types of chromosomes, and 1 plus 22 equals 23. So 22 different autosomes plus one sex chromosome type equals a total of 23 different types of chromosomes. So N is the number of types of different chromosomes. All right, so the diploid or 2N state describes the possession of two homologous chromosomes for each type of chromosome. So within each type of chromosome, whether it is any one of the 22 autosomes or possibly it is a sex chromosome, for each type of chromosome, we're going to have two homologous chromosomes. That's where the pair reference comes to. Okay, so for each type of chromosome, we are going to have a pair of homologous chromosomes. All right, and so if you have a pair of homologous chromosomes for each of your total of 23 different types of chromosomes, all right, you are said to be in the diploid or 2N state. All right, because if you have two sets of chromosomes for each type of chromosome, the total number of chromosomes is going to be the number of types of chromosomes times 2. Okay, so again, therefore, the diploid state in humans is a total of 46 chromosomes. You have 23 pairs of homologous chromosomes, or an homologous chromosome for each type of chromosome. And how many different types of chromosomes do we humans have? That's right, we have 23 different types of chromosomes. Of those 23 types of different chromosomes, we have 22 different autosomes, and then we also have a pair of sex chromosomes. The haploid or 1N state describes the possession of one chromosome for each type of chromosome. All right, Therefore, the haploid state in humans is 23 chromosomes. Okay, so 23 single, completely different types of chromosomes. So in this case, for each type of chromosome, okay, so for each type of chromosome, there is only one representative chromosome. Okay? There are no homologous chromosomes, all right, within the category of each type of chromosome, all right? So there are no pairs of chromosomes for each type of chromosome, as we find in the diploid state, all right? So these concepts can be confusing at first. It is worth it, however, to slowly review the concepts above one by one and commit them to memory. Remember, this is not a process that can be accomplished in one sitting. It will likely take several reviews of this material and different memorization techniques to finally internalize it. As always, you can always come see me in the office hours so that we can draw pictures of the various states and discuss their significance. So speaking of pictures, the next two slides contain karyotype representations of the diploid and haploid states in the human being. Alrighty. So this is a human karyotype that shows the diploid or 2N state. The numbers below the pairs of homologous chromosomes designate the number of each chromosome pair. Okay, so all of these numbers designate a special specific type of chromosome. More specifically, the numbers are referring to the different autosomes. All right, the sex chromosomes usually are not referenced by a number. Instead, they are often just referenced by their single individual identities. All right, instead of being referenced by a number. But if they were referenced by a number, they'd probably be referenced by 23. All right, so the numbers are giving reference to the 22 different types of chromosomes among the autosomes. All right. So remember, within each chromosome of each homologous chromosome pair, there are two 100% identical sister chromatids. Recall that the sister chromatids are closely aligned side by side. They are connected by a kinetic core at the centromere region of each chromatid. If these terms seem foreign or confusing, it would be a good idea to go back to the Chapter 2, Part 2 lecture and review them once or twice more. All right. So speaking of sister chromatids, let's reinforce that sister chromatids are 100% identical copies of the same chromosomes. They are not homologous chromosomes. In this picture, each homologous chromosome actually possesses two sister chromatids. So within each of these homologous chromosomes, within each pair of homologous chromosomes, so here we have, all right, the pair of chromosome type number 1, okay? This is one homologous chromosome, and then this is the other homologous chromosome. Within each individual chromosomes, okay, there are actually two sister chromatids linked together by a kinetic core at their central centromere regions of DNA. Now, right here, you can't really see the sister chromatids as being distinct within each homologous chromosome, but maybe if I can just insert some general lines to show the outlines of the shapes of the sister chromatids, it will better help you visualize what's actually going on. Now this one's going to be a little more complicated since it's twisted. All right. So here you can see that I'm drawing in these very, very slender X shapes, okay, which represent those X-shaped chromosomes that are composite of two sister chromatids, all right, as we have reviewed before. All right. So it's important to realize, however, the presence of sister chromatids in a single chromosome, which I'm telling you is the case in this diagram, or in this picture rather, that the presence of sister chromatids in a single chromosome does not have anything to do with the terms diploid or haploid. That's very important that you realize this. The terms diploid and haploid have absolutely nothing to do with whether or not there are sister chromatids. All right, diploid and haploid signifies whether or not there are homologous chromosomes for each chromosome type. So that for the 22 autosomes, each chromosome type is signified by a specific number. The sex chromosomes, a type of autosome, or a type of chromosome rather, not an autosome, but a type of chromosome themselves, they are usually signified again by their individual identity X or Y. All right. And as you can see here in this specific diagram, that is the case. For each individual type of autosome, we have two chromosomes. They are in fact homologous chromosomes lined up side by side, okay, by the technician who arranged this karyotype. All right, and then down here with the specific type of chromosome, the sex chromosomes, here we have an X and a Y chromosome lined up together side by side. And by the way, just to jog your memory, what's the gender relevance of this state of having a homologous pair of sex chromosomes of an X and a Y? All right, right. That means that this person, whoever they are, has the male gender, and if this person were to possess instead a homologous chromosome of the sex chromosomes of a full X chromosome, of a second X chromosome, all right, then that individual would instead be a female. All right. So again, the term diploid signifies the possession of two homologous chromosomes for each type of chromosome. The term haploid signifies the possession of only one chromosome for each type of chromosome. All right. So now let's go to the haploid karyotype. So in contrast, this karyotype shows the haploid state. There is only one chromosome for each type of autosome. Okay, so for each number here, which represents a different autosome, we only have one representative. Okay, here's only one chromosome, of which inside of it there are two 100% identical chromatids. Same thing here for autosome number 2. There is a single chromosome in it, which are two identical sister chromatids, but in either case, we're missing the homologous chromosomes. They're just not there. All right. So how many chromosomes do we have in total? Well, it's just 1 times the number of types of chromosomes that we have, and let's see, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 22 distinct different types of autosomes, and then 1 distinct type of sex chromosomes. In this case, it is the X chromosome, for a grand total of 23 different types of chromosomes. And so the total amount of chromosomes that we have is 1 times n, which is the different types of chromosomes, okay, and 1 times 23 equals 23. So here the haploid number, all right, for this haploid state in the human being would be 23. So finally, we can finally discuss mitosis. Mitosis is the process of cell division that occurs in somatic cells, your trillions and trillions and trillions of somatic cells. All right. If you recall from high school biology, conception begins with or when a sperm fertilizes an ovum or egg. Both the sperm and the ovum possess a haploid set of chromosomes. Therefore, both the sperm and the ovum contain only one chromosome for the 22 different autosomes and only one of 2 possible sex chromosomes. In other words, they only possess one chromosome for each of the 23 different types of chromosomes. All right. When the sperm fertilizes the ovum or egg, both of these cells' haploid chromosomes combine to form, yes, a diploid set of chromosomes. Okay. And a diploid set of chromosomes is one for each type of chromosomes. We have 23 different types of chromosomes, 22 different autosomes, and one set of sex chromosomes. We have, for each of those 23 different types of chromosomes, we have 2 different chromosomes. These chromosomes are homologous chromosomes. They are homologous pairs of chromosomes. So you have a homologous pair of chromosomes, or a pair of homologous chromosomes is a better way to say it, a pair of homologous chromosomes for each different 23 types of chromosomes. All right. I know I'm repeating myself over and over and over again, but the reason that I'm doing that is because I want to set an example for you. So to help you realize that the only way to really commit this stuff to memory is to say it to yourself over and over and over again, write it down over and over and over again. Suddenly, stop everything you're doing and try to test yourself as to whether you've really committed these terms and concepts to memory. All right. And then as time goes on, we won't have to do that anymore. They'll be part of us, and we can move on and use them in applications for much more interesting and specific purposes. All right. So anyways, what I'm getting to here is that when the 2 haploid chromosome sets from your 2 gametes, the sperm and the egg, come together in the process of fertilization to create a zygote, those 2 haploid chromosome sets are combined to create a new diploid chromosome set in the zygote. All right. And then what happens after that? All right, in humans, the zygote will be the first cell of an entirely new and unique human being. All right, at one time, we all started out as a single cell or zygote. However, it is the process of mitosis, okay, by which the zygote divides and divides and divides into a newly forming embryo. These divisions can continue hundreds of times as the newly growing human embryo develops. As the cells of the growing human embryo divide, many will begin to differentiate. In other words, many cells will take on specific roles. As time goes on, more and more cells will differentiate after many cycles of mitosis. These processes of growth and differentiation via mitosis continues outside of the womb as well, and even into the early adulthood for some cell populations. After the developing human being is born, any damaged cells will be addressed with division of both differentiated and undifferentiated cells. By the way, the term for undifferentiated cells is stem cells. So these processes of regeneration, or the replacement of damaged cells with newly formed cells, is also accomplished through the process of mitosis. Okay, so here for the total purposes here for this type of cell division. We have growth, all right? How do we get from one single cell as zygote to 100 trillion or so cells? Um, that requires a whole lot of growth. Within the process of growth, you have a lot of differentiation, cells taking on more specialized roles, and then sometimes those cells become damaged and they need to be replaced, all right? In which case they need to be regenerated from either differentiated cells or undifferentiated cells, aka stem cells. In that case as well, it is mitosis, which is being employed as the means of cell division. All right, so mitosis occurs in the context of a cell cycle. The cell cycle, of which mitosis is part of, is called the mitotic cell cycle. All right, it is important to realize that somatic cells undergo active division in the mitotic cell cycle and only spend a small fraction of their time in the process of actual division, aka mitosis. All right. As we've already noted, the process and time period where an actively dividing somatic cell is undergoing cell division is called mitosis. All right. In contrast, the time period in which the somatic cells are not undergoing mitosis and thus are not undergoing active division is referred to as interphase. So interphase is the phase or time period between mitotic cell divisions. And just think about the word interphase, okay? The suffix inter simply means between, and the word phase means time period, okay? So interphase is the time period between the actual process of mitosis where cells within the mitotic cell division are dividing, all right? So both mitosis and interphase are part of the mitotic cell cycle. All cells which are in lineages of cells that are involved in reproducing themselves via mitosis are in the mitotic cell cycle. When you are in the mitotic cell cycle, you are either actively dividing via mitosis or you are between cycles of mitosis in which you are said to be in interphase. All right, so don't worry, we're going to look at a schematic of this, so hopefully these terms will all start to settle into their proper place. All right, but I just wanted to go over it first with you, just to introduce some of these terms and the relationships between each other. Again, somatic cells in the mitotic cell cycle spend the vast majority of their time in interphase. All right. The interactive diagram on this slide has a wheel or pie chart that depicts the stages of the mitotic cell cycle in proportion to the relative amount of time that they occupy during a typical 19 to 24 hour or 19 to 25 hour cell cycle. You should keep in mind that this timeline is idealized. It does not represent every cell in the body. While there are many exceptions to this timeline, it is, however, a reasonable place to begin. Okay, so the first phase of interphase, all right, is termed G1. G, by the way, means growth. G stands for growth. The somatic cell will spend the majority of interphase in G1. During G1, the cell will enlarge. The interactive diagram on this page shows a representative cell with a single unwound, here we go. Okay, so this is a single representative cell with a single unwound chromosome in it. Okay, the unwound chromosome, as we have discussed before, is said to be in the form of chromatin. All right, a combination of an unwound or relatively unwound DNA molecule combined with a whole bunch of proteins that help maintain it. The black line within this linear and decondensed or unwound chromosome symbolizes the stretch of DNA that will eventually become known as the centromere, okay, as the chromosome condenses. Remember, the centromere is the central part of DNA, all right, and the relevance of it is that the kinetochore, okay, which connects two identical, 100% identical sister chromatids together, at their respective identical centromeres, right? Okay, so it's these areas right here where the kinetic cores will eventually attach to sister chromatids together. We'll go over that later. Right. So the second phase of interphase is termed as S phase, all right? S stands for synthesis. During S phase, all the chromosomes, 46 total chromosomes, will undergo an exact replication. This replication process takes place in the same way that was described in Chapter 2, Part 2. The strands of the original double-stranded DNA molecule composing each chromosome separate from one another at hundreds and thousands of different sites. Do you remember what these sites are called? All right, so right here I show you that the original double-stranded DNA molecules for this particular chromosome are separating from one another. In the red represents the addition of new strands. All right, and remember that these new strands are added by DNA polymerase, okay, and they are complementarily base paired to each of these separated original strands. All right, and in that process, we are able to begin to create two 100% duplicates of what once was one single chromosome. Okay? So basically, we're making an absolute 100% duplicate copy of this original chromosome right here. Okay? So do you remember what these sites are called? These sites where the DNA replication begins hundreds to thousands of times? Right, they're called origins of replications. Okay. Another thing is you know, remember DNA polymerase, the enzyme that's adding nucleotides, okay, to the new strand, so that the nucleotides lined up with nucleotides of the two original strands, all right, in a complementarily base paired fashion. Do you remember the directions by which the DNA polymerase is adding new nucleotides to the new growing strands shown in red here? Right, they're always added in the 5 to 3 direction, okay? So if one of these original adult strands or parent strands rather, if one of these original strands, let's just call them strands, if one of these original strands of the double-stranded original DNA molecule are headed in the 5 to 3 direction, okay, then the other original strand will be in the 3 to 5 direction, okay? In any case, DNA replication via DNA polymerase will be always accomplished in the 5 to 3 direction. So if the two original strands are already facing in opposite directions, the actual replication itself at each of these origins of replication is actually going to be occurring in opposite directions. All right, and as this happens, these loops get wider and wider and wider and wider and wider, and eventually they all run into each other. Okay, and as a result, this entire single strand, okay, of DNA representing a single chromosome is completely 100% identically copied, okay? So after the DNA molecules of each chromosome, all right, are completely 100% replicated with identical copies, S phase is going to give way to the third phase of interphase, which is called G2. in G2 rather, the cell prepares to initiate mitosis. In this diagram, you can see the two identical copies of the linear unwound chromosome still in the form of chromatin. Both the DNA molecules making up the two identical unwound copies of the chromosomes are in red and blue. What does this coloring represent? We'll look back at the coloration of the origins of replication in S phase. So the two new identical copies of the chromosome are the product of a semi-conservative mechanism of DNA replication. The original strands of each DNA molecule are colored blue, while the newly created strands of each DNA molecule are red. The semi-conservative mechanism of DNA replication was covered in the Chapter 2, Part 2 lecture. Please go back and review that if you feel that you're still on shaky ground with that. All right. So now we have two identical copies of this original chromosome that we started off with. Another thing, I just want to reach this point really quick. This cell does not have a nucleus to it, okay, just for the sake of simplicity. Another thing that this cell does not have is it does not have all 46 chromosomes, okay? This process that we just outlined, okay, from G to S to G2, in reality, in a human being, would be happening for all 46 chromosomes, okay, including each homologous pair of chromosomes within each type of chromosome. All right. This would be happening for each individual chromosome. Okay. We'd be making a complete identical copy during S phase. All right. So now we're going to head into mitosis. The yellow highlighted region represents the process of mitosis in its entirety. With respect to the wheel, however, notice that mitosis represents a very small fraction of the entire mitotic cell cycle. So specifically in mitosis, the two identical copies of DNA for each chromosome are coiled and coiled and coiled into the condensed version of the chromosome that we are all familiar with. Remember that only one chromosome is shown here for the sake of simplicity. Each of the identical copies of DNA becomes the different sister chromatids of the chromosome. By the way, as the book states, a condensed chromosome that consists of two sister chromatids, which represent identical copies of the same DNA molecule, is called a bivalent chromosome. So here we have the two identical copies of the same chromosome winding and 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 winding. Okay, and eventually each copy of the same identical chromosome becomes the two identical sister chromatids. Okay, and remember the sister chromatids are attached at their central DNA areas referred to as centromeres by this special protein here called the kinetochore. All right, and then all together, okay, we see this classic X-shaped chromosome structure referred to as a bivalent chromosome. Okay? So during mitosis, the two sister chromatids separate from one another and migrate to opposite poles of the cell. This process is called chromosome segregation. So here we see that the two sister chromatids have now separated from one another. The cell is still connected, or I'm sorry, the cell is still unified as one cell, but the sister chromatids of each bivalent chromosome are separating apart. All right. And this process, as we said before, is referred to as chromosome segregation. All right. And it's important to know that when the sister chromatids separate from one another, they are both henceforth known as daughter chromosomes. All right, so now these sister chromatids magically get a new name. One is a daughter chromosome, a whole brand new, fully designated chromosome, and the other one is also a daughter chromosome. When they were connected before, they were both sister chromatids of the same bivalent chromosome, but then during mitosis, they break apart at their kinetochores, and from henceforth, they are known individually as each being sister, or I'm sorry, I'm messing it up myself, each being daughter chromosomes, okay? They magically get a brand new name, okay? We're going to reiterate this all again when we go through the very specific processes of mitosis. This is just a big general overview right now. All right. So it's important to know that next the actual cytoplasm of the cell divides. All right. And so that the daughter chromosomes from each bivalent chromosomes are now separated from one another and contained within their own separate daughter cells. This process of separation of the cytoplasm, by the way, is referred to as cytokinesis. We will go over that shortly. Okay? It's important that I reaffirm that each daughter chromosome only contains one single copy of the DNA for that particular chromosome. All right? When each daughter cell enters interphase, so now each of these daughter cells are going to enter interphase. This one will go into its own interphase. All right. Each daughter cell only contains chromosomes with only one copy of their DNA molecules. All right. Then when each of these cells enter S phase again, this one single copy for each chromosome will be duplicated as two 100% identical copies for that same chromosome. And then when we go through mitosis again, we'll be back with a bivalent chromosome with two sister chromatids, each sister chromatid representing 100% identical copies for each type of chromosome. Or not each type of chromosome, but for each individual of the individual 46 chromosomes. All right. So you can see that the cycle continues on and on and on, just as outlined above, producing more and more genetically identical cells. All right. So throughout the entire mitotic cell cycle, the total number of different types of the total number of different types of chromosomes stays the same. Thus, the somatic cells retain the diploid state throughout the entire cell cycle, the entire mitotic cell cycle. The only aspect of the chromosomes that changes in their condensed or wound versus decondensed or unwound states, and the number of identical copies of each chromosome. I'll say that again. The only aspect of the chromosomes that changes is their condensed versus decondensed states and the number of identical copies for each chromosome. Okay, and we go back and we look at this diagram and we see that's all that's happening. That's really all that's changing as we cycle around and around and around the mitotic cell cycle. All right, we go from condensed chromosome to decondensed chromosome. All right, this is a single copy of the DNA for the chromosome, and then we switch to two identical copies of the same DNA molecule for each chromosome, and then finally we go back into the condensed state, and then we break apart, and then we go back down into the decondensed state, now containing only one copy of the DNA for each chromosome. All right, that's all that changes, but all the way through, we always retain a total number of 46 chromosomes. All right, within those 46 chromosomes, there are 23 distinct types of chromosomes. For each different type of chromosome, there are 2 homologous chromosomes. That all remains the same throughout the entire mitotic cell cycle. All right. So one thing that has been happening here that we haven't really spent a lot of time talking about is this switching between going from the decondensed state, okay, that you find in interphase, or the decondensed state of chromosomes that you find in interphase, and then transitioning into the condensed states that you find during mitosis. So that's one thing that I want to check out very quickly. And I apologize, I know we're going in a slightly different order here as is presented in the notes. All right, so basically what's going to happen is you are going to be represented with this same slide after we go over the next two slides, and we will just move on together from that point on. All right, but all of what I have said is still the same as it is written in your notes of your PowerPoints that go along with this. Okay, so now let's take a specific look at this process of condensation of chromatin during mitosis. Okay, so during interphase, the double-stranded double helix of DNA exists in a form referred to as chromatin. Each chromosome is a separate continuous double-stranded molecule of DNA. Keep in mind, however, that during S phase of interphase, a completely identical copy of each chromosome's DNA molecule is made. Chromatin is a complex of both the DNA molecules of the different chromosomes and proteins specialized for maintaining and organizing the DNA. Among these proteins, it is important, or there is an important group of proteins named histones. 8 histone proteins form groups that are referred to as octamers, around which the DNA molecule is wound and wound around many times. All right, so let's try to get to that point. So from here we have our single-stranded double helix DNA molecule, and we have it winding upon itself in tighter coils. All right, and so now we see these tightly coiled single strands of DNA then wound like spools of thread around these octamers of histones. Okay, so after the DNA is completely wound around one octamer of histones, it is then wound around another adjacent octamer of histones, and then it is sequentially wrapped in a similar manner over and over and over and over again. Okay, as a result, as I said before, the DNA molecule ends up being spooled like thread around a series of histone octamers. Each of these spools and its respective octamer is referred to as a nucleosome. All right, so this is a nucleosome. And then this is the next nucleosome. All right. So each chromosome may have anywhere from several 100,000 to millions of nucleosomes. That's a lot of spools of thread when you think about it. So in chromatin, the nucleosomes are then wrapped into pipe-shaped helical coils called solenoids. The solenoids are then wrapped into loops. So now let's look at these. So then these adjacent nucleosomes are wrapped around each other in a helical manner themselves, and we call the pipe-like shapes, or the pipe-like continuum of DNA that results from this, refer to this as a solenoid. All right. The solenoids are then wrapped into these big loops. All right, and then this is the form of DNA. Okay, as you have it in chromatin. All right. During prophase, the chromatin begins to condense even further, okay? The solenoids coil upon themselves, and then those coils coil upon themselves. Eventually, the successive coiling results in the classic X-shaped chromosome, or the bivalent chromosome, that you have two identical copies of DNA molecules. Each chromosome is completely 100% replicated during S phase of interphase. Right? So again, during S phase of interphase, we have this same DNA molecule completely 100% identically copied. Okay, and as this 100% identical copy is wound upon these octamers, and then the octamers, or the nucleosomes rather, are wound upon each other, and the solenoids are then wound upon each other, and those coils are coiled upon each other, and those coils are coiled upon each other. You end up with a whole nother identical sister chromatid. All right. So eventually we end up with two identical sister chromatids, okay, which are bound together at their respective centromere regions by a kinetochore. All right. So again, one of the identical strands of DNA are coiled like this, okay, up to this point within chromatin. And then we go through prophase. It starts coiling and coiling and coiling and coiling upon itself to create this highly regular and predictable structure of a single sister chromatid. And likewise, at the same time, another completely identical DNA molecule, okay, which was synthesized from this DNA molecule here during S phase of interphase, is going through the same process of winding and winding and winding and winding and winding, so that it forms an identical sister chromatid. All right, and then the two sister chromatids find each other and are bound at the hip by the kinetochore, and you end up with what we recognize as the bivalent chromosome. All right, does that help? You're starting to get this? If not, come see me about it. It isn't too bad. Once you get it, once you get the basic logic of it, it shouldn't be that bad. The first part is just starting to tackle all the structures and the names, but you go over it next time, or you go over it enough times and you're going to start to put it together. So all right, let's look at another representation of this process from another textbook. So here we have a nucleotide made up of a nitrogenous base, a sugar, and a phosphate group. All right. Oh, what's that from? Oh, I want to get rid of that. Well, that's left over. Let's see if I go back, maybe it will. Okay, yeah. Good. I have no idea what I just did, but I'm glad it's gone. Okay, so this textbook's rendition of the organization and coiling of DNA starts a little bit earlier, where we're actually constructing the DNA molecule from its basic subunits, which are nucleotides. So now we're connecting one nucleotide to another nucleotide. All right, and now we are orienting the nucleotides from opposing strands with one another, so that the different nitrogenous bases are associated with each other through these very specific hydrogen bonds, which are the basis between the very specific complementary base pairs between the nitrogenous bases of the nucleotides of the opposing strands of the DNA molecule. All right, and so now we are converting this molecular structure of the double-stranded DNA molecule for this highly schematic idealized version, okay, that basically looks like a railroad track, okay, where the rails are just alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and on and on and on and on. And the same thing's going here over on this rail. And then the ties of the track, okay, are representing the complementary base pairs of the different nitrogenous bases. All right, recall this. And another thing is, I'm just going to quiz you really quickly on this. If you recall that if you have a single ring to the nitrogenous base and it's characterized as a purine, as a pyrimidine. Pyrimidine, remember, big name, small molecule, big name. And then if you have a double ringed nitrogenous base, then that nitrogenous base is categorized as a, do you remember? As a purine. And it so happens that complementary base pairs only occur between a pyrimidine and a purine, or a purine and a pyrimidine, okay, giving this nice unified diameter between the two strands of the DNA as they interact with one another. More specifically, the pyrimidine cytosine is only complementarily base paired with the purine guanine, all right, and then the pyrimidine thymine is only complementarily base paired with the purine adenine, right? And these individual colors are representing the specificity in this complementary base pairing between the nitrogenous bases. Okay, and actually they have the color code of the specificity listed up here, where cytosine is shown as yellow, guanine is green, thymine is red, and adenine is blue. It's a little bit different than the color scheme that I gave you earlier, but in the end it really doesn't matter what colors we assign to these different nitrogenous bases. But anyways, I go over that just to quiz you on it, and if it's still something that you don't feel comfortable with, then going back to the Chapter 2 Part 1 lecture, or I'm sorry, rather the Chapter 2 Part 2 lecture might be a good idea. So now here we have the double helix of the double-stranded DNA molecule, and now this double helix is going to be spoiled around these octamers of histones. Okay, and all together we have one nucleosome here. Okay, and then here we have another sequential nucleosome here, and then on and on and on and on. All right. This is the pattern of DNA organization that you find in chromatin, the decondensed state of the DNA molecule of each chromosome. Then what happens is as we go into prophase, these coils of DNA will coil around each other, and those coils will coil around each other, and then those coils will coil around each other into this highly complex and elaborate structure known as a single sister chromatid. All right, that sister chromatid is joined by its entirely identical sister chromatid, okay, which represents this entire identical DNA molecule, which was obtained during S phase of interphase. All right. So again, all of this right here, the DNA winding, all right, going from the chromatin level of organization to the chromosome, or bivalent chromosome level of chromosome organization is occurring during prophase of mitosis, the first phase of mitosis. All right. All of these other organization patterns are exhibited in interphase. All right, so now we're going to go into mitosis in a little bit more detail, but I wanted to give you this basic overview of the big picture purposes and organization and time frame of mitosis before we went into all these details, mainly so that when we gave reference to a lot of these different terms involved with the big picture overview, we won't lose track of seeing the forest through the trees, so to say. All right. So we'll take a little break just for the sake of getting these videos up within about an hour time frame, so we can all get them up onto YouTube. I'm going to end here, and we are going to continue with chapter 2, part 3 in an additional lecture. All right, well, I'll see you soon.